Welcome back once again to my educational channel on biology for the KSSM syllabus and also for any other syllabus, biology syllabus, where it is relevant. So in this video, I'm excited to start a new series of educational videos on questions related to each chapter in your biology syllabus. Okay, so we'll start off with Form 4 Chapter 2, which is Cell Biology and Organization. And let's test ourselves after we have studied the content in the textbook and the notes. Let's test ourselves in order to obtain success or achieve success and achieve a good grade like A+ or A in our biology exams. So let's begin. So in this test, the total number of marks is 15 marks. There are 6 objective questions and 1 structured question. So let's start with the objective questions. Each question is worth 1 mark, right? So let's try the questions before we discuss the answers. Okay, take it like a test. So the first question, I will read the question and then pause for 20 seconds for you to find the answer. After that, if you still haven't found the answer, you can pause the video and spend another 30 seconds, okay, or 1 to 1 minute to find the answer. That's the maximum amount of time, okay? That is 1.5 minutes per question. All right, let's start with this first question. So I'll read the question. Density of organelles against the type of organelle, right? So we have lysosome, mitochondrion, and ribosome. The diagram shows a bar chart of the density of different organelles in cell Z, which is probably cell Z, right? So you have to know how to read this chart, bar chart. Now, what are the different answers? A, epithelial cell of artery, B, cardiac muscle cell, C, bone cell, and D, pancreatic cell. Okay, so I will pause for 20 seconds. Right, so if you still haven't found the answer or come to a conclusion on the answer, you can spend another 30 seconds on it, 30 seconds to 1 minute on this question. That's the maximum amount of time. All right, so you can pause the video, try the question, and now I'm going to give the answer. So the answer is B, cardiac muscle cell. Because the cardiac muscle cells have a high density of mitochondria. As shown in this diagram, this cell has a high density of mitochondria. In fact, for cardiac muscle cells, they have about 5,000 mitochondria per cell. All right. So this high density of mitochondria is needed so that more energy can be produced by using the mitochondria. Okay, through cellular respiration, the energy is produced. Okay, cellular respiration in the mitochondria produces this energy for contraction of heart muscles. Okay, so cardiac muscles are the muscles of the heart, and they are very hardworking. They are always contracting throughout life. So they need a lot of mitochondria to produce that energy for muscle contraction. Whereas these other cells, okay, do not require that much energy, all right, compared to the muscle cell that contracts. Now let's look at question 2. Small cylindrical components that exist in pairs in animal cells, made up of a complex arrangement of microtubules. The organelle described above functions in A. Intracellular digestion, B. Synthesis of enzymes, C. Cell division, or D. Transport of glycoproteins. So if you haven't found the answer, you can pause the video first, right? Right, let's look at the answer. C is the answer, cell division. So from the description here, we know that the organelles mentioned here are the centrioles, because it says they are small cylindrical components that exist in pairs. So there are 2 centrioles in each cell. Normally there are 2 centrioles, and centrioles are only found in animal cells, not plant cells, right? Made up of a complex arrangement of microtubules. So this is the description for centrioles, right, as shown here. So they're made up of an arrangement of small tiny tubes, all right? So this is a simplified diagram here. And so if they are centrioles, what is their function, right? Now centrioles form spindle fibers during cell division in animal cells, all right? And we'll study this cell division called mitosis, okay, and meiosis in the later chapter, okay? So they function, or they are active, and they play a role in cell division. The answer is C, okay? Next question, number 3. Which organelle is not correctly paired with its function? A. Endoplasmic reticulum synthesizes proteins. B. Golgi apparatus or Golgi body modifies and packs proteins. C. Lysosome breaks down bacteria. And D. Ribosome joins amino acids together. So 20 seconds from now. Right? The answer is A. Endoplasmic reticulum does not synthesize proteins. So this function is wrong. So this organelle is not correctly paired with this function. Now, what is endoplasmic reticulum involved in? So it's involved in the transport of substances, okay, like proteins. Proteins are transported by the rough endoplasmic reticulum, and lipids and glycerol are transported by the smooth endoplasmic reticulum, okay? This is the rough endoplasmic reticulum, and this is the smooth endoplasmic reticulum, okay? So for proteins, what are the organelles that synthesize the proteins? It's actually the ribosomes, or the ribosomes that are attached to the rough endoplasmic reticulum, or ribosomes that float, are suspended freely in the cytoplasm. So you look at this word, this function, ribosomes join the amino acids together, all right? So when they join amino acids together, amino acids are the basic units of proteins. So when they join amino acids together, they'll form the proteins. They synthesize proteins, okay? So this is correct, and C is also correct. Lysosome breaks down bacteria. B is also correct. Golgi apparatus modifies and packs proteins. So make sure you know all the functions of the various organelles and cellular components well. All right. So endoplasmic reticulum does not synthesize proteins, and this is the job of the ribosome, the ribosomes. So the rough endoplasmic reticulum transports the protein. Okay, it doesn't synthesize the protein. So this is a common mistake made by students. Now, apart from that, for smooth endoplasmic reticulum, they synthesize and transport lipids and glycerol. They can synthesize the lipids. But the synthesis of proteins is the responsibility or the function of the ribosomes. Okay. Now let's go on to number 4. The diagram above shows a unicellular organism. Which structure prevents the cell from bursting in fresh water? 20 seconds. Right. So you have to choose among these 4, right? Parts A, B, C, D. Okay. So the answer is C, right? So what is C? C is the contractile vacuole. As you can see from the pattern, the structure, it's shaped something like a flower. So you can recognize that this, you can identify it as the contractile vacuole in Paramecium species, right? So this cell is the Paramecium species cell. Now, so what does the contractile vacuole do? It excretes excess water by contracting, and when it contracts, the excess water is removed from the cell. It's excreted to prevent the cell from bursting due to excess water. Okay, so water is always diffusing into the cell by osmosis from the fresh water, and the vacuole will then be filled with this water. Its job is to, it will expand until maximum size, and its job or function is to contract suddenly and excrete the excess water out of the cell. If not, Paramecium species may burst due to the entry of excess water from the environment by diffusion. Okay, now what is A? A is the plasma membrane, and then B is the food vacuole, and D is the macronucleus. Question 5. The diagram shows an amoeba species cell. Structure Q is used in 1. Locomotion, 2. Reproduction, 3. Respiration, 4. Nutrition. So pick the right combination of choices. Right, let's look at the answer. Answer is A, 1 and 4. So first we have to identify structure Q, which is a pseudopodium. So it's used in locomotion. Locomotion means movement, all right? And it's also used in nutrition for feeding, all right? So during phagocytosis, the pseudopodia are extended out to engulf the food particle. So a food vacuole will be formed, and then the enzymes from lysosomes will digest the food in the food vacuole. Okay, so this pseudopodia, pseudopodium, is used in locomotion and nutrition. So it's 1 and 4. A is the answer. All right. Now let's go on to number 6. Which of the following cells contain structure X? A. Epidermal cell, B. xylem vessel, C. sieve tube, and D. Spongy mesophyll cell. So the answer is D, spongy mesophyll cell. So this spongy mesophyll cell is found in the leaf, all right, below the palisade mesophyll cells. And these cells have X, which is the chloroplast. So you have to identify that X is a chloroplast. Okay, right. So when these cells have chloroplasts in them, is because they can these cells, to enable these cells to absorb sunlight. Okay, the chloroplast will absorb sunlight because chloroplasts have chlorophyll, the green chlorophyll, to absorb sunlight, and so the cells can carry out photosynthesis to produce organic food substances for the plants. Okay, now epidermal cell is for protection. It covers the surface of the plant, and then xylem vessel is a cell that is dead and hollow, has a hollow center to transport water and mineral ions. Sieve tube is for the transport of organic food substances, okay, in the plant. Right, we've come to the structured question. So for this structured question, the total number of marks is 9 marks. And the time taken to do this test or this question is 12 minutes, around 12 minutes. Okay, that means for each mark is around 1.5 minutes. Okay, so do try the question yourself first, and take out a pen and paper to write down the long answers, the answers for this subjective question. Okay, then later we will discuss the answers and you can mark your own answers. All right, so apart from that, do underline some of the keywords in the questions, like functions, characteristic, all right, provide support, and so forth when you're answering the question. Okay, right, so now you can pause the video and then try the question, and after that we will discuss the answers together. Right, now let's discuss the answers. Question 1, diagram 1 shows a plant cell. A, name cellular components J, K, and L. So J first. So for J, we see that it has these C-shaped structures that are parallel to each other, and there are vesicles at the side here. So this is the Golgi apparatus, right? And then K is the inner line here. So it's not the cell wall. It's the membrane that is beside the cell wall, next to the cell wall. And this is the plasma membrane. Okay, so if you write cell wall, it's wrong. So unless the outer line here is labeled, that is the cell wall. Okay, the outer line is the cell wall. The inner line refers to the plasma membrane. Now L. For L, it doesn't have the vesicles at the side here, so it's not Golgi apparatus, and it doesn't have any ribosomes attached to it. So this is the smooth endoplasmic reticulum. Okay, so make sure that your spelling is correct. If your spelling is wrong, you won't get that 1 mark. So make sure the spelling is all correct, and the total marks are 3 marks. 1 mark for each label. Now B, state 2 functions of structure L in the cell. Structure L is the smooth endoplasmic reticulum. So the first function is that it transports glycerol or lipids. So the /9 means you write 1 is good enough. You want to write both is okay also. All right, either one's accepted. In fact, for the first function, you can also write L synthesizes and transports glycerol lipids. But here I just put the word transport because the main function is transportation for the endoplasmic reticulum. So the second function is L carries out detoxification of drugs or metabolic byproducts. Okay. Now, so when I underline all these terms, it means that these are the keywords, keywords that the examiner will look for to give you the answer. Okay. So if you don't write detoxification, you just write L makes the drugs not poisonous. This word detoxification is not there, then you will lose that 1 mark because we want this particular keyword. So remember, for answers in biology exam papers, you make sure that you write down that you can identify the keywords, write keywords and write it down in your answer. Okay, so explain one characteristic of structure K that is important for its function. So underline the word characteristic. K is the plasma membrane. It's important for its function. So note the function first. The function of K is to regulate or control the substances that move into the cell or out of the cell. Okay, so to do this, it has one characteristic, that is, it is semi-permeable, partially permeable, or selectively permeable. So textbook use the word partially permeable, and in chapter 3 of the textbook, okay, chapter 2, the word partially permeable is used. Chapter 3, the word selectively permeable is used. And sometimes generally it's called semi-permeable. Right? It's described as semi-permeable. So any one of these three can be accepted. Right? Make sure you have the term here, the special term. Okay? These special terms. Any one of these. Okay? So if you write K is not very permeable, then it's not correct already. Okay, we must have this term, these keywords. Now, secondly, you have to explain what it means. It's the meaning of the word explain. So you've stated the characteristic, now you explain. So what do you mean by semi-permeable? It means that it only allows free movement of certain molecules across it. That means molecules that are smaller in size, like glucose molecules, okay, or water molecules, and prevents or limits the movement of other substances. Okay, you can use the word prevent, it's easier. Prevents the movement of other substances or other molecules that are bigger in size, like starch molecules. They cannot pass through the plasma membrane. Okay. So D, how does organelle M provide support to a plant, especially to a herbaceous plant that has no wood in it? So there are 3 parts to this answer. Now 2 marks only. So the first point that's compulsory is that we must state that water diffuses into the cell by osmosis and fills M, which you identify as the vacuum. So sometimes you identify the structure, you can get marks. Sometimes maybe not. So, but it's good to identify first that M is the vacuum. So here I mentioned M, which is the vacuum, right? So the main point here is that in a dilute solution, water will diffuse into the cell by osmosis and fill this vacuum. Water will diffuse into the cell by osmosis when the external solution is more dilute or hypotonic, meaning that it has a higher concentration of water, environment has a higher concentration of water than that in the cell and in the cell, okay, in the vacuum. So water diffuses into the cell by osmosis when the cell is placed in dilute solution and or is surrounded by dilute solution. Then the vacuum will expand, P2 here, vacuum expands or M expands, and when it expands, it will push against the cell wall. So what pressure is created? A force called Turgor pressure is created. Now I didn't write this in because I've mentioned the word cell becomes turgid. But if one you can write in, when the vacuum pushes against the cell wall, okay, on all sides here, it will create the Turgor pressure that makes the cell turgid, that means firm and hard. And in this way, collectively, the cells will give support to the plant. So you write cell turgid for the third point. Give support, no more marks, because it's already here, provide support to the plant. So no more marks for this, but it's just to link it all together. Link the answer with the question, right? So P1 must be in. Water diffuses into the cell by osmosis and fills the vacuum. Then either P2 or P3. All right. So total here is actually 2 marks. Okay. So now we have discussed the answer. Try and mark your paper to determine how many marks you got for the structured question, right? So all these questions are important in the SPM. Now I'll be giving more tests, okay, for each chapter, because we've only tested a few subtopics, right? So we will see how it goes, and I hope that you have derived some benefit from trying these questions. Now let's look at your score. Okay, so total up all your marks and let's see how the score is given. What are the remarks for each score? Okay. Right, so here is a score chart to give you an idea how you fared or performed in this test, right? So for a score of 14 to 15 marks, converted to percentage is 93 to 100%, and this is an excellent performance, and the grade is A+. 12 to 13 marks, equivalent to 80 to 87%. Performance is very good, and it is a grade A. 10 to 11 marks, percentage, in terms of percentage, 67 to 73%. Performance is moderately good, and it's a B grade. 8 to 9 marks, percentage is 53 to 60%. The performance is moderate or average, is a grade C. 6 to 7 marks, percentage is 40 to 47%, and the grade is a D. And 5 and below, percentage is 33 and below, right? So for these two groups of people, okay, 6 and 7 marks and or below 7 marks, right? Do not be discouraged, as you can still improve on your grades. Okay, same with the rest too, all right, like average grade of C. So your grades can be improved with more practice, and make sure you get a good set of notes too. You can also go through my videos and jot down the notes from there, okay, as the notes are more complete, right? And don't give up, okay? So first of all, you have to get a good set of notes, and then you have to understand them, and after that memorize, okay, the facts. Finally, apply it by doing more exercises, okay? So the more exercises you do, the more practice you get, then you improve yourself, all right? So don't give up and learn from mistakes. These are the secrets of success, right? Thanks for viewing. Do share, like, and subscribe, right? And do comment too about these tests, all right? Whether you found it beneficial to you, okay? And whether it would be useful for you to have more tests in the future. Right. So take care and goodbye for now.